Well, thank you for the introduction. So we did this paper. It was published last year in PLOS Biology, and I'm very happy to talk about this huge effort today that we took in downloading 214,000 metagenomic samples and characterize the global abundance of antimicrobial resistance. So you might be a little bit familiar with the threat of antimicrobial resistance, or AMR. It is becoming and is a very global health threat. There's actually this study from 2016 that estimated by the time we reach the year of 2050, AMR is likely to cause 10 million deaths annually. There's actually, just last year, this study came out that estimated that in 2019, these authors found that almost 5 million deaths were associated with AMR. Of these, they could directly attribute 1.3 of them to antimicrobial resistant genes. And I really like this cartoon here, that it really well illustrates how easy it is for this transfer of antimicrobial resistant genes between microbes and pathogens and across environments and borders. There's also increasing evidence that antimicrobial resistance is not only a human health threat, but it actually is a global health threat in terms that it affects both the environment, animals, humans, and all these different kind of settings. So AMR really needs to be studied in this One Health context. So a big question of, okay, so how do we actually establish this study of AMR in all these different environments? So with this paper, we really wanted to ask, how do we create a collection of all metagenomic samples that spans the world across different environments, across different years? So of course, we could establish a large sampling collaboration. We could send me around the world a couple of times to just sample everything. But that would take a lot of time and a huge effort. But luckily for us, there's this really good practice in bioinformatics and sequencing that we want to share our raw data. And so with this paper, we wanted to utilize what is out there in public repositories. So what people put out on ENA, on GeneBank, or DDBJ. So we wanted to utilize this wealth of sequencing data that is available. And I really like this graph from ENA that shows how much data that is actually out there. So this graph illustrates the amount of sequencing data sets or runs that is available on ENA. It includes all kinds of sequencing data, so not only metagenomics. But so this graph shows that there's currently 26 million sequencing data sets currently available, and this number, it keeps growing. This corresponds to almost 50 petabytes of data. So there's a huge potential in utilizing all of this data. So in 2020, we wanted to do this. So we went in, we looked at the ENA, and we retrieved all the metagenomic data we could that was uploaded between 2010 and 2020 to ENA. That ended up corresponding to these 214,000 metagenomic samples. If I put some other numbers on it, it's almost 1.9 tera of reads, 442 tera base pairs, or if you'd like to talk about bytes, it's almost 300 terabytes of data. So it is a small subset of what is out there, but it's still a very large amount of data we could retrieve. So what did we actually do with this data? Well, of course, we retrieve the sequencing reads, and then we do this read-based mapping. So we trim them, we quality check the reads, and then we're aligning all these reads against two reference sequence databases. So we align them against the ResFinder database, which consists of antimicrobial resistant genes. At this time, there was a little bit more than 3,000 resistant genes in that collection. And then we also align them all against the Silver database, which contains more than 2 million reference sequences for the small subunit rRNA sequences. So we use this tool called KMA alignment. So you can see that we managed to map a lot of these reads against these reference sequences, but it's quite clear that we still have a lot of unknowns in this data. And meanwhile, while all of this alignment was running, I also did quite a lot of curation of this metadata, so trying to standardize a little bit, trying to see if it only has a GPS location, can I infer which country it is from, and so on. And so we stored all this in a database, of course. And from this database, I like to say that we are extracting count matrices. So count matrices is this idea that we are counting how many times we see different reads aligning to different references. And we can use this count matrix to characterize abundance both across the globe, across time, across different ecological settings, and so on. So this was all about what we did. Here is a little bit about the origin of all these metagenomic samples. We have the sampling source, the location, and the year. So for example, if we take a look at the sampling source, there is a lot of samples that are from human or from marine environments, but we also do have a lot that is from soil, from pigs, from chicken, from wastewater, and so on. So there's a lot of different environments represented. This is just the most frequent ones. There's a lot more in there. What's more interesting is if we actually look at the sampling location, so where the sample was physically collected from, we can see we have most of the world covered. Each dot is a sample. It's very clear we have a lot of different places, but of course, it's very clear that we have an overrepresentation of samples that are coming from North America and from Europe and also from Australia. We can also look a little bit about on the year when the sample was physically collected. We actually have samples coming all the way back from 1845, but we can see that a lot of these samples were taken in the last 20 years or so. So this is about where the data is from. We can also look a little bit about this huge sampling alignment effort we actually did. So here I'm visualizing the relative abundance of reads aligned to different resistant genes. This is colored according to which antimicrobial class we're talking about. I'm not showing the legend here, but I want to just illustrate that you can see that not all colors are distributed evenly. There's a lot of different stuff going on here. We can, for example, see that if you compare the European samples with the North American samples, the coloring are looking different. There seems to be something going on there. We can also look at the source a little bit. We can compare human pigs with marine samples. There also seems to be some very different profiles we can create from this kind of data. But I also said we mapped against the silver database. So I like to show this plot here that shows the relationship between the diversity of the microbes compared to the diversity of the resistance genes in these samples. So each dot here is a sample. There's a lot of samples, of course, but there's a tendency that a high microbial diversity means a high resistome diversity. But it becomes much more interesting once I color by, for example, the different sampling sources. That we can see that, yes, there is a lot of stuff going on. For example, we can compare pigs with soil samples. There seems to be a high diversity of the resistant genes in pig samples, where if you look at the soil samples, it's not all of them that have a high resistance diversity. Some of them, they have a high microbial diversity, but not really any resistome diversity. So there's a lot of patterns we really can explore with this kind of big data analysis. This is a very brief characterization of the data. There's a lot of stuff you can potentially answer with it. I like to say with this paper, this is a step towards utilizing everything that is uploaded and shared with the public. But since we're relying on what other people share, there's still some, I call them caveats, in terms of what you need to keep in mind if you want to use this data. So since we're relying on what other people share, we're also relying on correct metadata. It's only limited to what I can curate. So there's still several issues with metadata, unfortunately. And there's also, it's quite clear from looking at where these samples come from, there's a definitely large biases introduced in this collection, because we rely again on what people do, what they sample. We saw that there's a lot of human samples in there. Some environments are very well represented, some are not. And since we are working with the count of sequencing reads, Yeah, it's also very important to consider what the compositional nature of this kind of data, especially when you talk about what does a low read count mean. If I don't see any reads aligning to a reference gene, does it mean that it's not there? That we don't know. But it is a standardized data resort that allows or makes it possible to describe antimicrobial resistant genes, both in very local environments, but also at a very global scale. It is out there. I put it on Zenodo, where we're also working on making it a bit more accessible. So this paper was published in PLOS Biology, as I said. And I also like to add that we're actually doing this again as we speak. This time we want to do it better and bigger. We want to include the data that has been uploaded since 2020. We are expanding with other reference databases and with other tools. And we also, this time around, we also want to look at more at assembly-based methods. So we're also creating what I call targeted assemblies, which is this idea that we only want to create the contig and the scaffold if we see a resistant gene in it. And we are open to ideas, inputs. If you have any requests, want me to share other kind of data, also open to collaborations. If you see any questions you might want to answer with this data, reach out to me. And then finally, I just say thank you to several people, including Thomas Petersen and Frank Aarstrup, that have been my supervisors. And I will shamelessly plug my poster tomorrow that is showing how we actually use this data, showcasing different studies. And yes, so just say thank you.
We have time for some questions.
I thank you for the very interesting talk. How do you account for batching effect?
We haven't accounted for that. We just wanted to get everything, and then afterwards we are talking about how we can correct for all these batches.
Perfect. Now, regarding the methods, did you map your reads to any host, trying to filter them out, or just play with everything that you had?
No filtering of the host or anything.
And also no assembly, right? You use the quality control filters directly.
Yeah, so this paper is only about the read-based alignment, but we are looking into creating these assemblies now.
Perfect. Thank you very much.
You're welcome.
Hi, I have two questions. One question is, did you consider when you're looking at the resistome, the rarefication of the data?
No, we don't do that. We are instead looking at it compositionally. So we want to look at more whether the statistical test sets that the gene is there or whether there's a difference between different groups. We're not filtering on the counts themselves.
Oh, okay. Yeah, because sometimes you can look at rarefy the data,
Yes.
do the plots with the rarefication and then non-rarefication.
But then there's the risk that we miss all the interesting stuff.
Yeah. And then the next question that I have is, did you compare your results to the Metasub or Genome Tracker, any of those large consortium projects?
I haven't personally looked into it, but I have talked with some of the Metasub people. We have some of the data included, but it seems, since this is a much bigger scale, that I haven't really compared it yet.
Okay, thanks. I'll look at your poster.
Thank you.